Protein synthesis. Protein synthesis is the process of getting DNA to form proteins. Within this, two processes occur: transcription and translation. Transcription is the first process and allows DNA to be formed into messenger RNA, or mRNA for short. This is done in three stages. The first stage of transcription is initiation. RNA polymers binds to the promoter and starts at the template strand. This allows the DNA strand to unwind or unzip. When this DNA strand is unwinded, RNA nucleotides are able to be put in place. The second part of transcription is elongation. This is where C's and G's match up and A's and T's match up, but in RNA, T is instead U or uracil. The only part to be coded is the part that the protein needs. This will move down the strand and continue to elongate the RNA. This will eventually form the mRNA strand. Once formed, the third process, termination, allows the newly made RNA transcript to move out of the nucleus and into the cytoplasm. This is done when the strand hits a transcribed terminator. This signals the RNA strand that the sequence is finished and allows it to be released. This marks the end of transcription as DNA was successfully transcribed into mRNA. Translation is the second process in protein synthesis. To start this process, mRNA will attach to a small ribosomal subunit. When this attachment happens, the start codon should be read, typically AUG. This is done with a series of tRNAs. tRNA is the translator in this process and reads the three letters to make an amino acid. It is able to do this by bearing a specific amino acid at one end and the nucleotide triplet on the other to allow a complementary codon on the mRNA. Here we see the table used for the codon that mRNA has to send. Three of these signals is at the end to the sequence while AUG is the start. This table is directly read as the first letter being the left side, then the top side for a second, and the third level for the bottom. Once you match it up, you get the amino acid that is needed. tRNA comes in and reads the codon on the mRNA. By the anticodon region attached to the bottom of the tRNA, the nucleotide triplet can now match a specific mRNA codon and does its job of making that amino acid. tRNA that is translated leaves and another new tRNA comes to be translated. Each tRNA that comes makes one amino acid and adds to make a polypeptide chain. This process continues until a stop codon is reached. With the use of GTP hydrolysis, more polypeptides are able to be added. This is ultimately done with the use of the ribosome structure. The ribosome is laid out with an E, P, and A site. The P site holds the tRNA carrying the growing polypeptide chain. When the tRNA leaves, it does so through the E site. The A site will hold the tRNA carrying the next amino acid to be added to the polypeptide chain. The tunnel at the top of the ribosome is used to allow finished polypeptides to be released. Termination also occurs in translation. Once a stop codon is reached, the A site allows for a release factor. The release factor allows the hydrolysis of the bond between the tRNA and the P site and the last amino acid in the polypeptide chain. This ultimately will free the polypeptide chain and the protein is now formed. This will conclude translation and allow DNA to be made into RNA and thus into the protein, completing protein synthesis.